Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Kara Lynch. Listeners, Kara is a professor in the Department of Laboratory Medicine at the University of California, San Francisco. She is also co-director of the Core Clinical Laboratory at San Francisco General Hospital, overseeing chemistry and toxicology. And she is director of chemistry at UCSF Children's Hospital, Oakland. And today, listeners, we're looking forward to discussing Kara's career and her research, the impacts of her prior PCC-funded science, and also an innovative new research project focused on applying a novel high-resolution mass spectrometry approach for anti-doping analyses. So, Kara, we are so excited to have you with us. How are you today?
Great. Thank you for having me.
Well, thank you so much for joining us and sharing your expertise today. Perhaps we can start by learning a little bit more about you. So can you tell us a little bit more about yourself and how you got interested in research to begin with?
I actually started my life wanting to be a medical doctor, and I did research one summer when I was in undergrad and decided that research was the path for me. I liked exploring scientific questions and being able to do the hands-on work. So I pursued a PhD in biochemistry at the University of Wisconsin. And there I did basic science research and enjoyed my 5 years there, but wanted to do something that was more applied with my career. So I pursued a postdoctoral fellowship in clinical chemistry. It's a somewhat unknown field to PhD scientists. There are about 30 programs in the country that train PhDs to direct clinical chemistry and toxicology laboratories. I did a program at the University of California, San Francisco. That was in 2007 to 2009, and then was hired here to continue in a role as an assistant professor in the Department of Laboratory Medicine. So I have clinical responsibilities. So I oversee the clinical labs, as you mentioned. And the nice thing about that is I get to see where the needs are for technological advances and diagnostic testing, as well as toxicology testing. Get to be a part of a high production laboratory where we do routine mass spectrometry testing for toxicology. Get to evaluate novel technologies in my research lab that can be applied to the clinical lab. And that's how I translated that work into anti-doping work. There are a lot of similarities in the types of labs, of clinical labs and anti-doping labs, and the technologies that are used, as well as the substances that we're trying to detect. And in a clinical lab, we're trying to detect compounds that could cause patients harm. Most of my research is focused on small molecules and drugs. And the same is true for athletes. When the anti-doping, we're trying to identify compounds that can harm the athlete and give them potentially a competitive advantage as well.
Absolutely. We mentioned in your introduction that you have a variety of different roles, different hats that you wear in the work that you do. So how do you balance all of these different positions? And perhaps could you go into a little bit more detail on the scope of the research that you do?
Definitely. I mean, I think it is always a challenge to balance a lot of different responsibilities. And I'm sure many of you listening feel the same stress and pain of handling different roles. I think the nice thing about the work that I do is that it all informs, like my clinical work informs my research, and my research informs my clinical work. And a part of that is also teaching and mentoring, which is actually one of my favorite parts of the work that I do. And being a director of 2 different chemistry labs, my main lab that I'm at is at San Francisco General Hospital, which is a safety net hospital for the city and county of San Francisco. And we get a lot of unique cases here where we serve a predominantly underserved patient population. There is a high degree of substance use disorder among our population, so that makes it a fairly unique place to do toxicology testing. We also have a close collaboration with the Northern California Poison Control Center. So we get cases referred to us for toxicology testing from around Northern California. So balancing all of it, I think the work I do in the different areas inform each other. And by teaching in an academic environment, also we get a lot of great fellows and trainees that rotate through the laboratory and work in both the clinical and the research space. So it takes a team effort to get all this done. You also mentioned the types of research that we do in my research lab are predominantly looking at novel technologies to identify molecules and biological specimens. So that's a very broad topic. But a lot of my work has centered around mass spectrometry-based technologies and applying them to look at novel diagnostic approaches, looking at new biomarkers, looking at new emerging drugs and substances that could harm patients. And then also in the work funded through PCC, looking and trying to identify compounds in a better way that could harm athletes.
Absolutely. And I think you really highlighted well some of these overlaps between the clinical needs and the anti-doping needs with the technologies that you're working with. And I think this is a really valuable approach to have people with different backgrounds coming from different disciplines and applying their expertise in anti-doping. So I'm curious to hear, Kara, how did you first hear about the PCC and initially decide to apply for funding now over a decade ago?
I think it was around 2013, 2014. We acquired our first high-resolution mass spectrometer in our clinical lab, and we didn't start using it in our production lab in chemistry for quite some time, a few years, because really no one had done it before. So we were trying to figure out, how do you develop the method, validate the method, and make it where you feel confident in the results? How do you analyze the data? How do you train staff with maybe limited experience in high resolution mass spec to operate the instrument and do the testing and report out the results? There were just so many research questions. What's the best method to use? How should we acquire the data? How should we analyze the data? How should we come up with criteria that we use to determine if a compound is positive or negative in a biological specimen? How do we take advantage of the technology to identify emerging drugs and metabolites and novel and emerging doping agents? So there were a lot of questions about how we would utilize the technology. So I started thinking about funding that we could use to help with this. And a lot of the traditional funding mechanisms that are available to researchers don't actually fund method development and method validation and evaluation of new technologies. So I actually originally came across funding that was available through WADA. And then one of my mentors at the time had a close connection with Larry Bowers, who is emeritus on the scientific advisory board for PCC. I think he was also a key contributor to the formation of PCC. So he reached out to him and he said, hey, actually, you should consider the PCC funding. So that's how we learned about it, through our scientific network and connections with people who were working in the anti-doping space. And it seemed like a perfect fit. So at that time, we applied for funding just to really understand how the technology, high-resolution mass spectrometry, could be utilized in production labs like clinical toxicology labs, as well as anti-doping laboratories.
Very cool. And you were, of course, awarded that grant that you applied for that was really focusing on, like you mentioned, developing the methods and validating them for different, in the case of anti-doping, novel doping agents then back in 2014. So can you maybe share why this project was so important to conduct at the time in terms of focusing on these novel doping agents?
I'm going to draw on, again, similarities between clinical tox and anti-doping testing. I think we're challenged with sort of the traditional technologies that have been used through the years to detect these compounds that we're interested in detecting. So in the clinical tox world, we're using a lot of immunoassays to identify drugs or targeted LC tandem mass spec methods. Similar to the initial testing procedures for anti-doping analyses, they're designed to screen for the presence of a very chemically diverse group of prohibitive substances in biological samples with a high degree of sensitivity and specificity. But in some regards, a lot of those methods are, as I mentioned, targeted. So you sort of have your blinders up, right? You're just looking for those compounds that you know you should be looking for. So when we were embarking on this project, we first wanted to design a method that was broad spectrum, comprehensive test to be able to identify as many substances as we could within one method. So I think production labs end up having to run and maintain many different methods to detect all the different compounds that we want to see. So if we can streamline the number of instruments that we have to use, and streamline the number of methods we have to develop and validate and maintain, and make sure we meet all the regulatory requirements for that testing, it can be really helpful in a production environment. And also high-res, if you collect the data, and we collect the data in an untargeted manner, it allows us to go back and retrospectively analyze the data for compounds that may be emerging. So we first sought out to develop the method. We validated the method for a subset of about 250 drugs, metabolites, and anti-doping agents. We validated the method for sensitivity, specificity, limit of detection, all of your standard parameters that you do for validation of analytical methods in these types of laboratories. And then one of the big things I think had one of the biggest impacts is if you look up papers on high-res methods, you'll find good information about how people operate their methods, but you sometimes find very limited information on how laboratories identify what to call positive and what to call negative, or how to identify a drug and what criteria you should use for positive identification. The funding allowed us to do a very extensive study to look at what are the optimal criteria used for identification of these compounds and biological specimens with the highest degree of sensitivity and specificity. We also looked at some different library search algorithms for comparing acquired spectra in a biological specimen for a compound to a library spectra. And then we also used the funding to develop comprehensive libraries of mass spectra for compounds of interest. So that was some of the initial work we did with the initial funding we received from PCC.
Fantastic. And I think you highlighted high level what some of the results were, some of the things that you produced, but can you maybe comment on or maybe go into a little bit more detail on the impacts that these things, these libraries that you generated, these criteria that can be used to identify compounds within the results that you see, what impacts have those things had on anti-doping science?
Well, I think when we submitted the grant and received the funding, there really weren't many, if any, labs utilizing high-resolution mass spec. And when I wrote for this funding, in reviewing the literature, it's a more accepted approach. There are a lot more labs utilizing high-resolution mass spectrometry. So I think some of the initial work, the publications, raised awareness about the potential use and advantages of utilizing high-resolution mass spectrometry for doing a more comprehensive analytical testing approach for anti-doping as well as for clinical toxicology. So the technology has advanced. The use of the technology has grown. It has advanced. Others have contributed in this space as well. So I think the community is building of people that are utilizing high-resolution mass spectrometry for routine testing.
Wonderful. And as we mentioned earlier, you were recently approved for another PCC grant, this time focused on optimizing and evaluating a novel high-resolution mass spectrometry approach for anti-doping analysis. And we can get into some of the details, but can you first provide some background on why this problem that you're addressing with this particular grant is important for the anti-doping community right now?
So I think over the last 10 to 12 years, we've really learned the advantages and disadvantages of sort of the original generation of the first generation of high-resolution mass spectrometers that we used for clinical toxicology and anti-doping testing. And fortunately, the instrument manufacturers continue to make advances in the technology. So we recently acquired in our research laboratory a new type of liquid chromatography high-resolution mass spectrometer. It is a quadruple time-of-flight instrument, but has some advanced technologies built into it that allow us to do a lot of different things within the same platform, within the same mass spectrometer. So in the past, you maybe would have to have 2 or 3 different mass spectrometers to do what this one instrument can do. So I think it provides a great opportunity to evaluate this technology in a very systematic way to see how it might advantage anti-doping laboratories and anti-doping analysis. And so that was the reason why we submitted this funding.
Excellent. And can you maybe walk us through some of the details of this particular new research project? And perhaps we can start with just what are the specific aims? I know it sounds like a lot of questions that remain to be answered, but you always have to narrow it down to just a few. So what are the aims of this particular project?
To talk about the aims, probably have to get into the technology of the instrument a little bit. So for those of you that don't do mass spec testing, bear with me. I'll try to make it fairly straightforward. And for those of you that do, I also hope to provide enough detail for you to learn what we're planning to do. So when you're doing testing with mass spectrometry, your comprehensive drug testing, using a high-resolution mass spectrometer, we acquire the data in an untargeted manner. And when I say that, I mean that we basically are telling the instrument we want to identify compounds within a specified mass range. So for this work, we're only focusing on small molecules. So we cover the mass range that would cover all doping agents of interest and all clinical toxicology agents of interest. So the instrument first acquires like a full scan. So it looks for all masses within that mass range. It'll select the most abundant ions, send it to a collision cell, and systematically fragment each of those compounds to give you sort of a fingerprint or a mass spectra for each of those compounds that can then be searched against a library. So traditional approaches to this have used a specific type of fragmentation called collision-induced dissociation, or CID for short. So most of the methods that we use in clinical toxicology and anti-doping testing utilize collision-induced dissociation. But there are some limitations to this type of compound fragmentation, in that some compounds just don't fragment very well, and so you don't get a very good spectra, or you don't get a lot of mass ions within that spectra that provide really the specificity to know exactly what compound that is. And the new instrument that we're evaluating has a novel, tunable approach to doing fragmentation, and it utilizes electrons. So it's electron-activated dissociation, or EAD. This type of fragmentation can provide a very different way of fragmenting the compounds that we're interested in detecting. So we're looking at developing a library using EAD. So we're developing a comprehensive high-resolution library for EAD for both traditional and emerging doping agents and metabolites, and trying to determine its utility for anti-doping analysis. So we will compare the EAD spectra to the CID spectra and see in which situations this new way of fragmenting compounds can provide us better specificity in identifying anti-doping agents. So that's the first aim. And then obviously we will evaluate these libraries that we've developed and these methods in real specimens. So one of the advantages we have in being associated with the clinical laboratory is that we have thousands and thousands of samples that go through the laboratory on a daily basis. So we have access to a lot of biological specimens by which we can evaluate these approaches. So we will apply this to real samples that are positive for the compounds or the prohibited substances for which we're interested in evaluating. So that's the first objective and first aim. The second is that high-resolution instruments were traditionally thought to be less sensitive than the standard instruments we use in laboratories, which are tandem mass spectrometers. And those instruments are primarily used for targeted methods for identifying compounds. You have to know exactly what it is that you're looking for when you design those methods. And that was true, really, that when you evaluate high-resolution instruments and look at their limits of detection for all of the compounds you're interested in identifying, sometimes those limits of detection would be slightly higher or significantly higher than a targeted method for, say, 4 to 5 compounds. So you can imagine targeted method for 4 to 5, all of the settings and parameters and everything are going to be perfectly designed to detect those 5 things. And those 5 things may have all very chemically similar structures or behave similarly in the system. Then you go to a comprehensive method where you're trying to see as many things as you possibly can with very general settings and characteristics. You can lose some sensitivity. And another thing is, as ions travel through the mass spectrometer, or these compounds travel through and their fragments travel through, the traditional or the original high-res instruments sent them through in a continuous stream. And then in the time of flight tube, which is one of the parts of the analyzer, it would pulse these compounds or ions in packets through the time of flight tube. And as it would pulse them through, it would pulse at certain time points, but the ions were still traveling through at a continuous stream. And so you would lose ions that wouldn't go to the detector. So they made new advances to the instrumentation where you can trap those ions before they're sent through the flight tube to the detector. And we've shown in some of our preliminary data that this can significantly improve the sensitivity for compounds. It can also improve the quality of the spectra that are collected for the compounds. And all of this just adds to our ability to identify more compounds using the same method. But yet we haven't evaluated this technology for all compounds of interest for anti-doping testing. So we're hoping to do that with this funding and see how it performs, not just in laboratory stimulated situations where you're adding these drugs to blank urine, for example, but also in authentic urine specimens that we know are positive for one or more of the substances that we're looking for.
Very interesting. And it sounds like you are going to obviously play a key role in this project. Are there others in your lab group that will be contributing as well?
Yeah, definitely. So we have some postdocs that will be working on the project and a couple of research scientists as well.
Phenomenal. And we talked a little bit about the methods. Do you have expectations, I suppose, about what the outcomes or the products of this work will look like?
Yes, I would like to make the libraries that we generate available to the community, testing community. So that's one of my aspirations or hopes for the research, and creating maybe resources that are available to other laboratories, sharing what we know, obviously through scientific publications, peer-reviewed publications. Those are some of the goals and aspirations I have for the work.
And can you give us maybe a little bit of a layout of the timeline? When is the project going to start and what does that timeline look like?
We should be able to start soon. We're still working through some of the contracting between PCC and UCSF. So always more the administrative stuff. So once that's in place and the money is available, we'll get started right away. So we have been able to evaluate some of these new technologies with a smaller subset of drugs, metabolites, and doping substances. So we have some of the methods kind of in place and ready to go, and we're excited to get going and moving forward with a larger subset of compounds to see how the technology works for those.
Very cool. And then just to give our listeners a sense of, again, what the impacts of this potential work could be, can you share, you mentioned you want to make these libraries, you want to make them open to the community. How will that change anti-doping and potentially how labs are operating on a daily basis?
Well, one thing that we hope to explore in the future, too, is just how transferable libraries are between instruments. So if it's a library we generate on this specific instrument, it may only be useful for other labs that have the same instrumentation. But I think we could potentially show that this instrument is helpful and useful to the anti-doping testing community. That could lead to maybe their ability to utilize the work that we've done to help them purchase advanced instrumentation for some of the work that they're doing. I'm hopeful that we can show that one method could detect a large number of substances rather than having to use 5 or 6 different methods to detect the diverse group of compounds the anti-doping laboratories are required to test for. So that could lead to some workflow streamlining within laboratories, hopefully some financial savings. You can take 3 or 4 methods you have to maintain and put them all into one and only maintain one method. That could be really helpful. I also think that these advanced technologies offer a lot of opportunities for us as labs to stay ahead of the game, so to speak. There are always emerging substances, doping substances, novel substances, designer substances that are emerging, and the target's always changing. So I think the approach through the years in laboratories is traditionally to be more reactionary. So once we learn that something is emerging or maybe of concern, then is when the clock starts to develop a method, validate a method, implement a method. All of that takes time. With high resolution mass spec, you can retrospectively analyze data. You can do different types of data analysis. And that could allow us to potentially identify emerging substances more rapidly. And these broad spectrum tests allow us to add targets pretty easily to an already existing method. So we could potentially more quickly add targets and emerging substances to our methods rather than having to kind of wait and develop a full targeted method. So those are some of the things I think that could come of the research.
And I think every research project sort of gives rise to even more questions or directions or things that you can pursue in the future. So do you have ideas, Kara, at this stage of what some future directions would be for this particular line of research?
Yeah, I think that high-res mass spec has made its way into a lot of laboratories, but I think there's still opportunities for better utilization. So I hope that this work and what we publish can be utilized, the procedures can be utilized to help laboratories make decisions about instrument purchases and about method development and implementation in their laboratories. So that's one anticipated outcome. The newer instruments and newer technologies also offer a lot of different ways to acquire data. Right now, we use sort of a data-dependent acquisition. So we acquire spectra for like most abundant compounds within the sample. There are also data-independent acquisition approaches that we've evaluated through the years. There are hybrid approaches where we could do targeted analysis at the same time as untargeted analysis. There are other abilities where you can create inclusion lists into your method where you say, I always want to identify this subset of compounds, and exclusion lists where you can say, hey, we know that this subset of masses and compounds are always in the samples that we're analyzing. So you want the instrument to ignore those so it can focus more on the things that you are trying to detect, and that can improve the sensitivity. I think we're always going to need to be increasing our libraries and adding to our libraries so we can better identify compounds. So I think those are some of the future works that we could do after this work is completed, looking at some of the different ways of hybrid approaches within the same instrument to better detect the molecules and compounds that we need to detect with greater sensitivity and specificity. And I think also there's a lot of opportunities for informatics advances and shared knowledge. A lot of the software that we use to do the data analysis is provided by the vendors of the instrumentation that we use. There are some open source platforms, but I think given the era that we're in with a lot of technological advances and artificial intelligence and so forth, I think there could be a lot of advances in the next 5 to 10 years in how we take a sample, analyze a sample, and get the data output, and how we can analyze that output. So with high resolution mass spec, untargeted data analysis, you generate a lot of data for one sample. But right now, I'd say we're somewhat limited by our ability to analyze that data. And so we go at it with a lot of different approaches. So we analyze it in a targeted manner, or we analyze it in a suspect analysis manner where we say, well, we think maybe these compounds could be in there. We don't have a lot of experience detecting these compounds, but can we analyze the data and see if we have any clues to suggest that those compounds might be there? And really, we're limited in our ability to really do true untargeted data analysis. And those are just IT limitations. It's kind of like trying to find a needle in a haystack. So I think there can be a lot of advances in the future in regards to how we analyze the data we acquire with high resolution instruments. So we can apply that to be able to stay sort of ahead of the game and to be able to maybe even predict where the field is going with kind of emerging novel substances for doping.
Definitely, Kara. I think that all makes sense. And I think you alluded to what my next question was going to be, just some of the challenges, some of the limitations. So can you go into a little bit more detail? I think we made it sound easy so far, right? You just run the study, you get these beautiful libraries, you apply it to real samples. What is the reality in terms of when you're doing the science, there are going to be challenges and limitations that you have to work through. What do you anticipate with this project?
The biggest challenge is the chemical diversity of the different compounds that are on the prohibited list of substances. So labs now have many different methods that are used to detect all of those different compounds because they're so chemically diverse. High-res mass spec can allow us to pair together some of those substances of chemical diversity, but there are just going to be some molecules, say small molecules is what we're focused on, some small molecules that just don't behave well in the generalized parameters that we're using. So a single sample preparation method, a single chromatography method, a single ionization technique, or fragmentation approach may not be optimal for every compound. I think that's the biggest challenge. But on the bright side, the newer instruments allow us to do a lot within the same platform. So we can ionize compounds in different ways. We may have to analyze the sample more than once. We can also, as I mentioned, with collision-induced dissociation and electron-activated dissociation, we can fragment the compounds differently, maybe with different ways of pairing together, like scan modes within our method. So I think there's some things that can help with those limitations, but the experiments need to be done really to see if it works or if it doesn't work.
Absolutely. And then are there parts of this project that you think are particularly novel or particularly exciting right now?
I think the technologies I've mentioned. So first, EAD, a different way in which to fragment molecules. One example I provided in the grant is amines, a class of compounds. They don't produce very robust fragmentation patterns for routine collision-induced dissociation. So for example, methamphetamine, amphetamine, and a drug called phentermine, which is a drug that is used as like an appetite suppressant and can have some bad side effects. So it's suggested that it not be used very regularly or for a long period of time. So those are like three amines that all have very similar, if not the same structure. So two of them are actually isomers of each other, structural isomers. And when they are fragmented by kind of our traditional approaches now, they only produce 2 to 3 diagnostic fragments, is what I'll call them. In the grant, we're able to show that with EAD, you get very distinct and very rich spectra for amphetamine, methamphetamine, and phentermine, and can easily differentiate the three from each other based on the fragmentation pattern. And there's other compounds. One other compound I've provided in the grant is buprenorphine and its metabolites. Buprenorphine is a drug that's used for opioid replacement therapy for people who have a substance use disorder for opioids. And the fragmentation pattern is not very good for CID. So it really limits the specificity and sometimes sensitivity in identifying that compound. And with EAD, we get a very beautiful, robust spectra. So those are just a few examples. And I'm excited to see what it can do for a larger list of compounds on the prohibited list of substances for testing and some of the emerging compounds that we're interested in looking for.
Well, we are so excited about this project that you're working on, Kara. We're excited to learn more just about the collaborations. I think they're so important in science in general, but particularly in anti-doping science as well. So can you talk a little bit about how you and your lab collaborate with other stakeholders, whether it's local individuals or labs and groups that you work with, funding organizations like the PCC and others as well?
So in the clinical toxicology world, we work really closely with Poison Control Center, our clinicians that work in the space of caring for patients with substance use disorder, our addiction care teams. So that kind of informs our work in relation to how we do clinical toxicology testing. We also work very closely with the City and County of San Francisco and the Department of Public Health. We do biosurveillance monitoring for the City and County of San Francisco to kind of see what is emerging in the drug supply in our city. And then when it comes to anti-doping stakeholders and laboratories, attendance to conferences and giving talks and hearing the work that other people are doing and how our work can inform each other, how we can support each other and collaborate, that has been really helpful. Also, there are great conferences for mass spectrometry in laboratories, in the clinical laboratory, as well as anti-doping laboratories. And I've had the opportunity to interact with other people who have received funding through PCC. And I've been able to provide specimens to laboratories that are doing work from our sample set that we have here. So that's some of the examples of ways in which we've collaborated with stakeholders, like people who are working with PCC and have received funding through PCC.
Very cool. And I think, Kara, like I mentioned earlier, anti-doping science is really broad. It covers a lot of different areas. And we're excited to be bringing in scientists from all over the world who have different expertise. So can you share what your experience has been like really applying your expertise to problems in anti-doping?
Yeah, it's been very rewarding because, as I've mentioned already, many of the analytical problems are surprisingly similar. So in both clinical tox and anti-doping, we don't control really what comes into the lab. So new drugs appear, patterns of use change, the analytical methods have to be developed and have to evolve with them. So I think it's great that I can take my area of expertise and the environment that I'm in, which is more a clinical toxicology environment, and apply it to the anti-doping space.
Very cool. And one of the questions I love asking everybody we have on the show is, if we took away the barriers that normally hold you back, Kara, so if you had all of the funding, all of the resources, and feasibility wasn't an issue, and you could do anything, what is the one problem in anti-doping that you would most want to solve?
I would love to get to a point where we could perform truly comprehensive, highly sensitive, untargeted testing where we don't have to decide in advance what we're looking for. So even now with untargeted data acquisition, we almost all routinely then go back and analyze the data in a targeted manner because we're somewhat limited by the tools we have to really interrogate the data. I think it would be great if we could get to a point where we could truly perform true untargeted testing. If I could have access to every new novel mass spectrometry platform that becomes available, they really are advancing the technology that the diagnostic companies or the mass spec manufacturers, I should say. It would be great to have access to all those tools, but they are quite expensive. And some of our budgets don't necessarily allow us to purchase these amazing instruments. Even the research that's done on them can sometimes be somewhat limited. So unlimited resources, we would have all of the instruments to evaluate them all and compare them head to head and see which instrumentation, which technology, which method development type approach, which type of screening approach works best.
Very cool. Well, Kara, I love the sounds of this dream project. And if our listeners have been captivated and they're interested in learning more about you and the work that you're doing, where can they go to get more information?
You can find publications from my research group via PubMed or Google Scholar. You can look up my name. You can also visit my profile on the UCSF website. Feel free to reach out to me personally. I actually love to interact with other people working in this space or individuals that are interested in learning more. I do work in an academic environment where we're constantly training new scientists emerging in the field. So if you are a young scientist that's interested in this space, please feel free to reach out to me directly. My email is my name, kara.lynch@ucsf.edu.
Fantastic. Well, Kara, we appreciate you sharing those resources and your openness to connect with people in our audience here. And Kara, it's been such a pleasure to chat with you today and to learn more about you and the amazing work that you're doing.
Thank you. Thanks for having me.
Absolutely. Thanks so much for joining us. And listeners, always wonderful to have you here with us as well. Make sure you take a moment to check out Kara's profile on the UCSF website. Find her papers if you want to learn more. And listeners, if you haven't already, it would mean a lot to us if you could take a moment to rate our podcast and leave a review on your favorite podcast platform. Please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.